Hi, everyone. Welcome to episode 99 of Cell and Gene, the podcast. I'm Erin Harris. In this episode, I'm talking to Dr. Kenji Fujita, Chief Medical Officer at Atsena Therapeutics. Dr. Fujita is responsible for overseeing clinical and regulatory aspects of the Inherited Retinal Diseases, or IRD, pipeline, which includes 2 clinical programs. He and I talk through Atsena's approach to treating inherited retinal diseases, specifically X-linked retinoschisis, or XLRS, which is a rare disorder that affects vision and currently has no FDA-approved treatments. We discuss how Atsena is using novel gene therapy technology to tackle this significant unmet need, including an interesting innovation, what they call a spreading AAV vector that could overcome delivery challenges. Dr. Fujita also shares what it means to receive fast track designation from the FDA and how it could accelerate bringing this much needed therapy to patients. Plus, we break down the science of XLRS, what makes this disease so complex, and why targeting the RS1 gene is the key to restoring vision. If you're interested in the future of retinal gene therapy and how new technologies are changing the game, you won't want to miss this episode. And so here's my conversation with Atsena Therapeutics, Dr. Kenji Fujita. Enjoy. Hi, Kenji. Thanks for being here today.
Sure, it's my pleasure.
Good. All right. So talk to us a little bit about Atsena Therapeutics and your role as CMO.
Sure. So Atsena is a clinical stage company devoted to applying novel gene therapy technologies to unmet need in inherited retinal disease. The company was founded about 5 years ago by Shannon and Sanford Boy out of the University of Florida, and a lot of our technology is based on decades of work in their labs. We've made great progress in these past 5 years. We now have 2 programs in clinic, both of which are showing really promising results. So I'm the CMO, the Chief Medical Officer, and my primary responsibility is to direct our clinical development efforts. And as part of that, I oversee multiple supportive functions such as regulatory, patient advocacy, and safety. I'm also part of the executive leadership team, which guides corporate strategy and manages relationships with investors and business partners.
Excellent. Thank you. Okay. So XLRS is a rare inherited retinal disease that currently has no FDA-approved treatment. So I'd love for you to talk us through, provide an overview of the disease, tell us about its impact on patients and how Atsena is working to address this unmet need.
Sure. So XLRS is one of the more prevalent inherited retinal disorders. There are an estimated 30,000 patients worldwide. The disease is caused by mutations in the RS1 gene, which has a number of functions, not all of which are fully understood, but it's involved in maintaining structural integrity, maintaining fluid balance, and maintaining proper communication between the different retinal cell layers. It's an X-linked disease, which means that it affects males. Boys with the condition usually present in elementary school, and typically the teachers will notice an inability to focus on a blackboard or an overhead projector. They'll be referred to an optometrist or an ophthalmologist who will do a routine examination. And there they'll find a characteristic abnormality called a spoke wheel pattern, which is just what it sounds like. The next step is usually a more advanced technique called OCT, which is a technology which allows the doctor to get a better view of the retina in cross-section. And then this shows a very characteristic pattern of distinctive large cavities between the layers of the retina, which look almost like Swiss cheese. So very, very distinctive, hard to miss. Clinically, most patients have vision between 20/40 and 2200. And so what this means is that most of them are sufficiently impaired that they can't drive, but they're not legally blind. Fortunately, the disease is stable through most of middle age, although patients are at greater risk of retinal detachments, retinal tears, other trauma, so they are advised to avoid kind of contact sports. But eventually, by the 5th or 6th decade, you do start to see degeneration of the retina, which will ultimately lead to blindness. So even in patients with good baseline vision, there is an imperative to find treatments, both to improve the vision that they have, but also to prevent progression to blindness in the later years. And unfortunately, as you pointed out, there are no currently approved treatments. So Athena's approach is to go right to the source of the problem. We're looking to deliver a healthy copy of the RS1 gene to replace the defective copy. The way we do this is that we package the healthy gene in an engineered virus called an AAV vector or capsid and inject that complex right under the retina so that it can get into the photoreceptors and replace the defective genes. One way of thinking of this is think of the gene, the healthy gene, as kind of a business traveler out on a mission. And the vector is like a taxi that takes the passenger to the destination, which in this case is the photoreceptor. So the surgeon will go under the retina, inject a little bit of liquid containing the capsid, and then the capsid does the rest, brings the kind of seeks out the photoreceptors, injects the healthy gene to the photoreceptors, and we hope by this method restores normal function and improves vision.
Good. Okay. And thank you for that explanation.
Sure.
That's really helpful. So Atsena recently received fast track designation for XLRS gene therapy, the program. So talk us through a little bit about what does this designation mean for the development and regulatory pathway of the therapy? And then, on top of that, how will it potentially accelerate bringing it to patients?
Okay, sure. So fast track designation is one of the tools that the FDA has to help accelerate development of promising therapies targeted at serious or life-threatening disorders with significant unmet medical need. So what this means practically for us is that it gives us more frequent and easier access to the FDA to work out any problems or any issues with the development program and to streamline the process. But it also means is that when and if we get to the point where we're applying for marketing authorization, we will be able to apply piecemeal. In other words, submitting documents as the data come in on a rolling basis rather than waiting for everything to be ready and submitting all at once. And the advantage of this is that it allows the FDA to review on a rolling basis, gives the reviewers a head start, and can lead to a faster approval than would be capable otherwise. What this means for patients is that the FDA recognizes the severity of the disease and the urgent need for new therapies, and is devoting additional resources and time to working with us to get this thing to patients as quickly as possible, assuming that all the data are positive.
Good. Okay.
Yeah.
Let's talk a little bit about inherited retinal diseases. They present unique challenges, including precise gene delivery and durability of the treatment. So, what are some of the key scientific and clinical hurdles you've encountered to date, and how is Atsena addressing them?
So one of the biggest hurdles in XLRS is that in order to be effective, the RS1 gene has to be expressed in photoreceptors. And so if you look at the structure of a retina, it's composed of multiple layers of cells, with the photoreceptor layer buried under several layers of other cells. So to access that layer, you need to go and surgically lift the retina and inject the therapy underneath the retina. This is problematic in XLRS because remember what I mentioned about the Swiss cheese appearance. These patients have large regions of the retina that are structurally abnormal, that are more fragile than a healthy retina, and are not amenable to surgical manipulation. So it's very difficult to go in and take this approach. So others have tried to get around this problem by taking an intravitreal approach. What this means is injecting the therapy into the vitreous, which is the fluid in the middle of the eye, with the hope that enough capsid and effector will reach the deep layers and deliver their payload to the photoreceptors. These approaches have unfortunately not been successful because there are no known AAVs that are able to be injected intravitreally and reach the photoreceptors. So Atsena has the advantage of a novel technology which allows for safe subretinal delivery, even in patients with significant structural abnormalities, as are typically seen in XLRS.
Gotcha. Okay. I want to continue a little bit on the topic of AAV vectors. So as you've said, Atsena's approach to retinal gene therapy includes the use of novel AAVs. Talk to us a little bit about how your proprietary vectors differ from traditional AAVs, and then why are they critical for treating XLRS, and other IRDs?
Yep. So we're working on a number of new technologies in vectors, but I think the most important for this conversation is our spreading vector. So we've developed a novel vector which we call AAV.SPR, SPR for spreading. And the way this differs is kind of going back to the initial description of how the vector is delivered. The surgeon has to create a space under the retina and inject some fluid in a puddle or bleb. And just for safety and logistical reasons, that bleb can only encompass a very small portion of the overall retina. With conventional subretinal vectors, they're only able to deliver the gene to cells that are directly in contact with that bleb. So really only a small percentage of the retina, and that bleb has to be placed in the critical part of the retina. This is a problem for XLRS, again, because these patients have large parts of the retina that are just not accessible by this methodology. So we've developed a new technology, a new vector that is able to spread laterally across the retina away from the area where it's injected and to deliver the gene to cells that are remote from those blebs. And this is important in XLRS because even in severely affected patients, there are areas of normal structure that are amenable to surgical manipulation. And so with this spreading vector, we're able to deliver subretinally by targeting multiple small blebs to structurally normal areas of the retina, which can be safely administered, and then counting on the spreading nature of the vector to kind of go laterally and to deliver the gene to areas that are not accessible surgically but are critical for vision.
Good. Okay. So we are recording this after Rare Disease Month, but I think anyone who has, a rare disease or is caring for someone with a rare disease, every day is Rare Disease Day, which is the last day of February. And patient advocacy and engagement are very significant in rare disease research and development. And so how is Atsena working with the XLRS patient and advocacy community to ensure their voices are considered, throughout the whole development process?
So patient advocacy is an important part of our mission. We have a dedicated director of patient advocacy who works very closely with the various patient advocacy groups, as well as working with individual patients to keep them informed of our progress. And it's really important for us that we hear the voice of the patient through those channels. We've had a number of recent activities to enhance that. One is we recently launched a Voice of the Patient survey, which was sent out to patients and caregivers to help us learn more what the day-to-day experience is living with XLRS. We're planning to follow that up with a live focus group where we'll be able to communicate directly with these patients and caregivers and have more of an interactive dialogue to both learn about their experience and to communicate some of what we're doing internally. Finally, we've put together a number of webinars that patients and their families have been welcome to attend.
Excellent. Excellent. All right, let's talk future roadmap. So looking ahead, what's next for the program and what key milestones should the industry and the patient community be watching for in the coming months?
So this is a really exciting time in the program. We've just completed enrollment of part A of our study, which is the dose finding part, and identified a safe and effective dose, we think to be a safe and effective. We've started enrollment of our part B, which is our dose expansion, where we'll get more clinical data on that selected dose. So we are planning to present interim data at a meeting later this year. And we also plan to meet with the FDA to clarify what the next steps should look like and what sort of data package they need to see in order to move this forward to an approved therapy.
Excellent. Well, Selene and certainly I will be following along and looking forward to that information to share with our readers and our listeners. And listeners, that brings us to the end of this episode featuring Atsena Therapeutics' Kenji Fujita, MD, Chief Medical Officer. Kenji, thank you so much for your time. This was wonderful, insightful, and looking forward to working with you again.
You're welcome. It was my pleasure.
Good. All right, everyone. Thanks for tuning in. Be sure to visit cellandgene.com, and we'll talk to you soon.